Situated here. Nice to meet you and see all this enthusiasm for, let me get all this stuff out of the screen, that everything Brooke and Tanya are doing. I will first start by introducing myself. Let me just finish this. Okay, excuse me one second here. Well, that's good enough. All right, so very nice to meet everybody, and I just want to say that I'm very grateful to Brooke and Tanya for this opportunity to speak to the group. Um, I'll start by saying I am a paleontologist. I'm a vertebrate paleontologist. I also am an anatomist, and my work has been in a range of morphology or phenotypic work, comparative work about the tree of life. So I could describe myself as a systematist or a taxonomist. And in this talk today, I'll be speaking in very general historical terms. It's not a talk about hypotheses or about the value of methods. It's more a historical description of what's happened over my career and how MorphoBank came to be. So first, I'll just present what I plan to cover, and that is I'll start by talking about the importance of systematics. Now, this is probably something that many of you appreciate already, but just to give a little bit of my take on that. Then talk about how morphology is important to building the tree of life. We're living in a time where there are many kinds of data considered for the tree of life. So what is morphology's role? Modern morphological systematics is a field that needs the same kind of digital support and web support that other fields do. I'll talk about what has been done in MorphoBank, what you can do in MorphoBank, how I have used it in my own scientific career, and then talk about the need to pass things that are invented by scientists to the hands of capable supporting programmers and PhDs who can carry things on. So I'll start with the importance of systematics. When Science Magazine turned 125, about 20 years ago, it took a moment to pause and talk about what are the key things we can hope to discover in the next 100 years, which is a good thing for a magazine of this nature to do, to stop and say, where are we going as a scientific community? And I was glad to see that one of the questions and areas that they put on this list was the discovery of the tree of life. Will we get scientists to agree? Will we make progress in methods, in consensus in this area? So this is just to underscore that the work that we do as systematists is very important to science as a whole, and morphological systematics is important in that context as well. So morphological systematics is just building the tree of life. And this is an older diagram from the scientist Haeckel. So we are essentially trying to come up with a tree that explains the branching, the history of all of the species, both living and extinct. And this is done, of course, now with all kinds of data, from nucleotides to genomes to proteins and to phenotypes, including behavior, structure, morphology, etc. So why all these data? Why not use just one kind? Well, because parallelism and convergence are real phenomena. We don't have a Rosetta Stone of phylogeny, no matter how passionate any given scientist is about their data and what it means. There's examples of parallelism and convergence occurring in all these kinds of data. So bringing different kinds of data to the table helps us make a better tested tree of life. Also, it's important that to keep in mind that 99% of life is extinct. There's an excellent essay introducing the book Extinction and Phylogeny, which came out in 1992 with the editors Novacek and Wheeler. And they have a nice essay about the fact that looking at the relationships of living organisms today is a slice of life as a whole, and that 99% of life is extinct, and a portion of that has fossilized. So bringing in the fossils as direct evidence, which is often with morphology, is very important, again, to making a tree of life that is indeed really a tree of life. So, to look at where we see morphology and systematics in a context of contemporary work, here are two examples. If you're a user of MorphoBank and you've looked at the breadth of publications that have had their authors deposit data in MorphoBank, you'll see often the Journal of Vertebrate Paleontology and the Journal Cladistics. They're two of hundreds of journals that publish morphological systematics. But if this isn't your stomping ground, it might not, it might come as a surprise. But there are very commonly published matrices and trees based entirely on morphology or based on what we call total evidence or combined data, and they're coming out all the time. So it's just, I just mentioned this by way of saying that this is active, ongoing research that MorphoBank is trying to capture. So, to talk a little bit about what is involved in doing morphological systematics, I'll just put up this kind of flow chart of how we work as scientists day to day, those of us who build trees using morphology. So, if we start on the upper left, that's a skull of a fossil hominid. Could be anything, could be an impression of a butterfly, could be a living species that has morphology, but we observe directly with our eyes some aspect of the anatomy of these animals or plants or anything living. And then as we move to the right side of the screen, we translate that into anatomical terms. And immediately you can see that those terms are becoming digital. They're words that represent something about what we see, and then we represent them here in this case with discrete characters as zeros and ones. So we've moved from something being a visual and verbal observation to something being digital. Those digits then are assembled in the matrix that you see on the bottom left. And probably many of you are familiar with the common formats used in our discipline, which are Nexus or TNT, both invented by practitioners. And that's how we assemble the data that we observe. The species are typically in rows and the observations in columns. So that's just sort of a convention. Those matrices are then fed into algorithms and produce trees. So this is just the workflow that we typically do. Now, this might be a good junction to say that when I began to work on MorphoBank and to develop MorphoBank, I was in a world where science had become highly gene-based and nucleotide-based. So because of the medical need for that kind of data, there has always historically been much better database support for the gene side of biology. For morphology, there was less so, and there was nothing much for what's going on with the day-to-day collection of morphology. So it was in doing this workflow that I thought to myself, we need something better. We need databases and we need repeatability built into our science. So those points, which are in some ways on this slide right now, are, if we think about the development of those matrices and the way they have been used by scientists from the 90s, the aughties, the teens, and to the 20s now, collaboration is fundamental. More and more scientists that do morphological systematics are working as a team. Secondly, they build on prior data, and that is a good thing. They want to take a matrix that's published in a peer-reviewed publication and add data to it, change it, revise it, make it better, make it address the things that they observe. So that's important as well. And third, we are, over the course of my career, we've been in a phase of upping our standards of research replication across the board. So, for example, if the matrix that we saw on the last slide appears as a picture in a digital or paper publication, that's not of great use to another scientist who comes along. And I'm not joking that many of us have had to retype by hand those matrices because they haven't been found in databases. And that completely slows down the advance of science, regardless of what perspective you're coming from. Here are two of my colleagues, John Weibel and Eric Seifert, collaborating on a matrix, and you can see the expression of the man's hand on his head perhaps captures a sense of frustration that scientists historically felt when they were working in matrices that were single files on any given computer. Yet at the same time, they were trying to collaborate. So this may sound like, oh, such a simple thing that should be solved, but it's not been, it's been challenging to move the field in a direction where we have software that stops what one developer calls the pass the pumpkin problem, where you have a fancier term to put on that is version control, where no one knows if scientist A or scientist B has the final version, and you waste time trying to pull your collaboration together because you're all working at the same time and you all need access to a single copy of the data. So by putting matrices in a database, that solved that problem. And then by putting that database on the web, it allowed scientists who were either in the offices next door to each other or sitting next to each other or across the globe to collaborate. So if we add some of these points into this workflow, this red box captures some of the things that I was just saying. So when a peer review publication appears, the matrix that underlies the science should always be available not as a picture, but as an executable, computer readable Nexus or TNT format. Now, there was some headway into that in the 1990s, but MorphoBank has brought that, has emphasized that even more, particularly for morphological data. We need a good correspondence or perfect correspondence between what the words were that the scientists used to describe the characters and what was meant by the digital information. The excellent curators of MorphoBank, Tanya and Brook, are very familiar with the deposition of just a matrix, which is only half the information that a new scientist needs in order to repeat a morphological study. Um, Obviously, they should be publicly available. You shouldn't have to ask the scientist for his or her work once something is peer reviewed. It should be publicly accessible. And finally, because MorphoBank is its reason to be is to back up this important scientific work that's happening on morphology. There should be a way to record those visual observations, especially as we live in a digital world now where images are all digital and numerous, that we should allow scientists to put those into a database because they all won't fit into a peer-reviewed publication that enriches the work. So these motivations, these goals are totally in sync with the efforts, the social efforts in science to engage in responsible conduct of research. And I know this audience is familiar with the importance of that. And I just put up a couple of respected entities that have emphasized how key it is to do and have repeatable work. And we want morphology to be part of that. So what does MorphoBank do in this context? Well, if you look at this slide, what you see in the center is a matrix, but it looks a little different than the matrix that I showed you in the earlier slide. Instead of being just text and just digital information, it's a colored, enriched spreadsheet with pictures of the animals, pictures of the information that was used to make what's called a homology statement. That's the thing in the boxes, and then other information that enriches what the scientists had done, takes a better snapshot of their work. So for example, instead of putting just a 0 or a 1 in a cell and leaving it at that, on the top left, the scientist can add the text describing what he or she thought about the character information. There can be real-time record of the notes of who made the score if it's collaborative, what other information they might want to share on the upper right. They can put an image of what they think they're saying. For example, if you want to say that this particular species had an elongate jaw and you think that's better communicated in a picture, you can add that picture. That can be a CT scan, it can be a 2D image. For example, for my own work, I find this extremely helpful that I can go back to an image, a labeled image, and look at what I meant by something 5, 10, 15 years later. And finally, sometimes it's helpful to have a picture of the species that you're looking at, and that's also possible. And all of this is within a database. So the good thing about this is that a lot of times scientists find it hard to, at the end of a very demanding preparation of a peer review paper, to gather all their information for history to put in a database. But if you collect your information in a database while you are working, all you have to do is push a button and it goes out. So um, the very key emphasis is that MorphoBank is both a place to work and a place to store work. This slide shows the team that I collaborated with from about 2007 to 2017, when we worked on something that was called the Mammal Tree of Life project. And this shows the very problem that I was talking about before, that there's a real obstacle in place if you're working on a file-based version of the matrix that's located on a single desktop. You can't really work as a team. So if you put that matrix in a cloud, in a web application that's also a database, suddenly more than 25 researchers can work simultaneously. And this is what we did in developing the Mammal Tree of Life project. And perhaps this is a good junction to say that um, we're extremely grateful to the National Science Foundation for what was nearly 20 to 25 years of support for MorphoBank in different ways, through the paleontology community, through the biology community, through the computer science community. Have been various different entities contributing to the sustaining of the site, the rebuilding of the site, and we're extremely grateful for that, and it's made it what it is. So here is a slide from that project showing again the same kinds of things. When we did this Tree of Life project, we assembled one of the largest, maybe the largest matrix for morphology ever published. It has over 4,000 observations for, I think, around 75 species. And it was the product of the collaboration of about 25 researchers working over 6 years. And we of course built on prior data. We revised that. We added images of our observations and produced something that's been reused in different publications, rebuilt on, and was a very rewarding project, regardless of the debates about the science and what this means for placental mammal evolution. Those things are conversations for another day. But as a collaborative project, I think it was a great example of how software can serve collaborative teams. This project would have been impossible without MorphoBank, and MorphoBank itself grew greatly. So that paper is the one on the bottom right that was published in Science. So that was a very rich collaboration. But I've also published papers on the top there about just what it meant. So many of the points that I've touched on in this talk. The second author on that paper is Seth Kaufman, who's the owner of a company called Whirligig and was the primary computer scientist behind, computer engineer behind the development and the origin of MorphoBank. He was a steward of its health for 20 years. And perhaps it's a good junction to also thank Kensley Alphonse, who was trained by Seth and also did this. And also to say that when you work in biology, there are bioprogrammers who produce lots of interesting tools, but I am of the belief that those tools need to pass into the hands of professionals if we want to sustain them as a community. And that's where people like the Phoenix bioinformatics community come in and become so important, because as everyone knows, you can't leave a piece of software just sitting on the web and expect to find it working when you come back to it 2, 3, 5, 10 years later. The web is like a garden, everything changes, there's bugs and weeds, so you have to constantly sustain the health of that site. You have to rebuild the code. And those professional programmers like Seth Kaufman, Kensley Alphonse, and the whole Phoenix bioinformatics team have been essential to that happening. The other paper in PLOS was a collaboration with many scientists who are more gene-based, and we're trying to to create a tree of life that was a kind of constantly updating entity that was bringing in data from peer-reviewed papers and updating itself. And so we wrote about the best practices that are needed across the discipline so that data scientists can get the data. They don't have to go into the original literature and find, again, pictures on pages or obscure and confusing Nexus files that don't execute and various things like that. We laid out some standards that practitioners should follow so that they can do their best to leave data that are reusable. So I'll conclude by just saying that in my own practice as a scientist, in my own role as founding MorphoBank, encouraging its use, speaking about it, which is now almost over the last 30 years, I feel that it's important to pass a creation made by scientists to professionals who can sustain it. And that's where the incredibly important work of Phoenix Bioinformatics comes in. If they did not exist, they would not be able to sustain the important databases that we see on screen. And I think as a community, what we can do to help them is to, try to get our institutions to support this entity. The model that we use at MorphoBank, kind of like National Public Radio, where it's free for everybody, but those of us from institutions that can contribute do contribute, that we keep an eye on sustainability if we think these resources are really important. So thank you very much for your attention. It was a pleasure to speak to you and meet you, and I wish Tanya and Brooke the very best with their meeting.
Thank you, Maureen. Do you have time for a couple questions if anyone has any?
Absolutely.
If anyone has any questions, feel free to you can either type it in the chat or you can unmute.
It's not like a question. I was just like thank her for this amazing metrics. I'm using this metrics now in some of the whale matrices, and the pictures and the characters in this matrix is amazingly documented. So thank you.
Oh, my pleasure. And I'm so glad to hear that. I'm so glad to hear of scientists using the data there. And that's fantastic. And I look forward to seeing your publication that results from that. I see some things in the chat, but
Just praises
great.
about your work.
That's nice. Great. Well, it takes a village. All the people, the names I see in the chat were also, they've all done their part to help spread the word for this, to make this really, it's not about any one person. I mean, I've done some, but it it was important for my day-to-day work, so there was a real synergy there. But I'm glad to see it. It will only happen if people believe in it, and I'm really glad. I've always wanted to make it a community project, and Brooke, what you're doing, Tanya, what you're doing is really fabulous.
Thank you, Maureen. We do have one question that just came through in the chat. To what extent would you envision recommending or enforcing standards for coding phenomics, such as 0 is absent, whereas 1 is present? I know in my curation experience, sometimes 1 is absent.
Right. My feeling is those are questions, those are research questions for peer-reviewed papers, for that kind of thing. As a database, manager, I would not enforce it, because right now we're still in a phase of trying to get the community to participate. So there's always this balance between putting up standards and people bristling at the standards and saying, well, I don't want to contribute my data because I don't want to meet the standard that I disagree with. That said, I have learned from computer scientists have taught me that, you know, the more standards, the easier the curation. So just like Brooke just said, so it's a balance. I right now I feel like the scale is still tipped towards trying to get people to use the site. We're still in a generational shift where people are not always inclined to even make their work repeatable. But if you have a strong feeling about that and you have, you know, ideas, the places to bring those are to the Hennig Society meeting, to the Society of Systematic Biology. There's lots of meetings where those kinds of things that's what they do. So go there and talk about it, and maybe then you will become the spearhead for that change. And that would be, you know, we could debate the reasons why one or the other, how it doesn't matter scientifically, all those kinds of things, but those are probably conversations for another day. So my sense right now, my takeaway is that the lower the bar, the better, until we get really full-blown community participation, not to put Brooke out of work.
Thank you.